Okay, guys, so we're gonna try to test this out. This is gonna be a screencast on explaining a little bit of the mechanisms by which prokaryotes or bacteria regulate genes. This is something that you've seen already. It is something that you read in chapter 18, specifically section 4, but I want to go through it just really fast with you guys to make sure that you got this, okay? So we know why genes in prokaryotes or in eukaryotes are regulated. They're regulated so that both organisms can be super efficient at what they're producing. Genes, remember that the vast majority of genes code for proteins. A lot of them code for enzymes, so they're coding for the molecular mechanisms by which we do all of our biochemical processes. Okay, so all of these enzymes are coded for in genes. You do not want to code for enzymes, that are not needed at that moment, because that is inefficient use of your resources. So both bacteria, prokaryotes, and eukaryotic organisms have developed mechanisms to be able to regulate the transcription and translation of genes, and they do this based on signals. Okay, so you can, specifically we're going to be talking about gene regulation in bacteria, and you can turn off genes in bacteria, or you can turn on genes in bacteria. So it's a switch where it can be on or off, and this switch is particularly called an operator. So as you guys are going to learn very soon, and genes will be regulated in a bacteria in transcription units, which are not just one gene. It can be 1, 2, 3, 4 genes, and all of those genes are adjacent in the chromosome, and they're adjacent because they're usually coding for enzymes that work together for one particular process. So you want to turn all of those genes on at once if you want to go through that process. For example, the digestion of lactose is one operon that has a lot of genes that code for enzymes that all work together so that bacteria can digest that lactose. So all of those genes are together in this operon. Okay, so genes grouped together with related functions. You can have an operon that's inducible, or you can have an operon that's repressible. The words should already tell you something. If the operon is inducible, that means that the chemical signal that is used to influence the transcription and translation of those genes are induced. Those genes are induced by the chemical signal, so they're turned on by the chemical signal. If the operon is repressible, that means that the chemical signal actually turns off the transcription and translation of those genes. It turns off the operon. So if you think about it logically, this makes a lot of sense, because when do you want to turn on some genes? You would want to turn on some genes whenever the chemical signal is something that you need to break. So for example, in one of the operons that we've talked about already, the lac operon, you guys are going to hear that lac operon is an inducible operon. The reason why it's an inducible operon is because its function is to break down lactose. You want to have that operon turned off. You don't want to be producing any enzymes to break down lactose if there is no lactose present in the environment. So the lactose, in this case, in the operon, is actually allolactose, which is an isomer of lactose. But the chemical signal that lactose is present is going to turn on the lac operon so that bacteria can start producing enzymes to break down the present lactose. So it'll induce the operon. If you are actually talking about things you want to not break down, but you want to create, you want to have an anabolic process, anabolic process, then you want the chemical signal to be a repressible, you want the chemical signal to be a repressor to that operon. What does that mean? If you've also talked about this and learned about this already, the tryptophan operon, TRP, the trip operon. Tryptophan is an amino acid. All prokaryotes need to produce this amino acid. So when there is this amino acid in the environment, the prokaryote can simply get it from the environment and does not need to produce this amino acid through anabolism, right? They do not need to because they have it in the environment. So when tryptophan is in the environment, this tryptophan is going to become the repressor of this operon, so that the operon that codes for enzymes to create tryptophan is shut down, since you do not want to produce tryptophan at that point, since you can get it from your environment. So this is all about being efficient with your materials. Before I show you these lac operons and this tryptophan operons, basic ideas, which you've already talked about as well, and you've answered these in different questions. I just want to make sure they're clear. Promoter is the section in the operon where the RNA polymerase is going to bind. So it's the RNA polymerase binding site. You've probably heard about this in the eukaryotes with the promoter, the very famous promoter called the Tata box. Okay, prokaryotes also have promoters, and they're simply the site in which the RNA polymerase is going to bind. And if the RNA polymerase is able to then move towards that segment of DNA, then it's going to be able to start transcribing messenger RNA. Okay, so it's that RNA polymerase binding site. The operator is the switch. Okay, so the operator is actually where the transcription and transcription in this case, the transcription of that DNA or of that operon is going to either be switched on or switched off. And how is it going to be switched on or switched off? With the presence of regulatory proteins. One particular regulatory protein are repressor proteins. If there's a repressor protein in the operator, it does just that. It represses RNA polymerase from actually moving in the DNA and transcribing that DNA into mRNA. So it represses transcription. Okay, so let's see the repressible operons. The example that I showed you, tryptophan. Okay, so what happens right over here is that here you're going to have the here you're going to have the promoter. Okay, the promoter is where the RNA polymerase is going to bind to. Regardless of anything, the RNA polymerase can always bind to the promoter. The problem here comes from the next site of DNA, which is the operator. The operator can either have a repressor bound to that operator, or it can be free of the repressor. In this particular case, there is a lot of tryptophan. So what does that mean? Tryptophan is going to be something that's called a co-repressor. So molecules of tryptophan, amino acids of tryptophan, are going to actually bind to a repressor molecule. And when that happens, that repressor molecule is going to bind right over here to this operator and block the operator. Since the operator is downstream from the promoter, then the RNA polymerase will no longer be able to move from that DNA and to transcribe mRNA. It will not be able to do this. So since it can't do this, it's not going to produce any of these enzymes, and that's okay because these enzymes are there so that the bacteria can produce tryptophan. And since the bacteria does not need tryptophan, since the environment is filled with tryptophan, then it's okay to repress this operon. What happens when there is no tryptophan in the environment? The bacteria needs tryptophan. It is a nutritional necessity of the bacteria. The bacteria will die without tryptophan. So since there is no tryptophan in the environment, there's going to be no co-repressor molecule, no tryptophan, which is called a co-repressor. So nothing is going to bind to the repressor molecule. What does that mean? The repressor molecule is not going to want to be active. It's going to want to be inactive. What does that mean? That means that the repressor molecule is not going to bind to the operator. It's going to be pretty much just in the cytoplasm, okay? Not bound to the operator. So that means that the RNA polymerase that is in the promoter can easily start transcribing that DNA since it has nothing blocking it. Once it starts transcribing that DNA, all of these enzymes are produced, okay? And you can start producing tryptophan, and the bacteria is going to be able to survive. So that is a repressible operon. If you're talking about an inducible operon, you're talking about the lac operon, which is the prokaryote's ability to break down lactose. Okay, so if there is a lot of lactose, we're gonna have to think about it a little bit in an opposite way than tryptophan. Okay, so lactose is not the molecule of lactose, unlike tryptophan, which is a co-repressor. Lactose is not a co-repressor. Lactose is an inducer. So what does that mean? And that means that when there is a lot of lactose, okay, lactose is going to bind to the repressor molecule, and when it binds to the repressor molecule and activates that repressor, that means that the repressor can no longer bind to the operator, so it falls from the operator and it's just chilling here in the cytoplasm. Okay, it's no longer bound to that operator, it falls away from that operator, and that means that RNA polymerase, which is bound to the promoter, can start moving downstream and transcribing all of these genes into enzymes. What are these enzymes going to do? These enzymes they're enzymes that their function is to break down lactose. So their enzymes going to start attaching to these lactose and start breaking these down. Okay, so this is very important because as they're breaking down the lactose, the amount of lactose is going to decrease, right? So when the amount of lactose decreases, what does that mean? Less lactose to induce this entire operon. So when the level of lactose reaches 0, no lactose can attach anymore because you don't have any lactose anymore. You've broken everything down. No lactose can attach to the repressor. So that means that the repressor is now active. The repressor can move towards the operator, bind to the operator, and repress that RNA polymerase from moving towards the downstream from the DNA and transcribing that DNA. You're repressing this. You're no longer allowing the RNA polymerase to move. But that's okay as well because you no longer need any of these lactose breaking down enzymes since you no longer have any more lactose since you've broken everything down. So this is similar to things that we've talked about before of positive and negative feedback loops so that you can reach a balance and you can reach homeostasis, okay, and you can use your materials effectively. One last thing that we want to talk about this operon is that operons don't only need to be switched on and switched off, like turned on and turned off. They also have a volume switch. So you can either accelerate the transcription of a operon, or you can have a transcription of an operon that is pretty low. Okay, so how can you do this? Besides having this operator, you're also going to have a section or a site within this operon that's called the activator. So let's see if we've got any of these activating genes, activating sections here. Okay, so bacteria need sugar. Lactose is sugar. So lactose is a good thing for bacteria because it can break it down into galactose and glucose and use that as sugar. Okay, but the bacteria definitely, and many organisms definitely prefer just pure sugar. We prefer glucose. Okay, lactose takes some energy to break down and do stuff with. So whenever there is glucose in the environment, even if there's lactose, you want to be using the glucose and you want to be breaking down less lactose because it is, again, bad use of your energy. It takes energy for these enzymes to break down the lactose. Why would you have to do that when there's already glucose in the environment? So glucose, the presence or absence of glucose, can be a volume switch. Okay, it can either increase the transcription of the lac operon or it can decrease the transcription of the lac operon. How can you do this? This you do through something that you've already talked about as well, which is cyclic AMP, adenine monophosphate, adenosine monophosphate, is a form of ATP without the other two phosphate groups. Okay, so this was, if you recall, in your signal transduction pathways lecture, this was called the second messenger. So it does a lot of things in your signal transduction pathways, and it's going to do a lot of things in gene regulation. Basically, when you have a lot of when you have very low glucose, there's not a lot of glucose in the environment, there's actually a lot of cyclic AMP, CAMP. Okay, so there's that inverse relationship. Very low glucose, a lot of CAMP. So a CAMP is an activator. What does that mean? It's going to activate, or it's going to bind to this catabolic activator protein. And once it binds to this catabolic activator protein, this entire complex is going to bind to something that's called the cap binding site, or what we can think about as the activator site. When this protein is in the activator site, what it kind of does is that it pushes the RNA polymerase so that the RNA polymerase can actually go a bit faster. And if the RNA polymerase is going a bit faster downstream from the DNA and transcribing all of these genes, then you can transcribe these genes faster. You can make these enzymes much faster. So what you can think about is the level of glucose. If glucose is very low and there is lactose present in the environment, then CAMP with the catabolic activator protein right over here is going to promote, it's going to increase the rate at which transcription is being held, and the enzymes for the breaking down of lactose is being produced. So you're going to increase that rate. Why do you want to increase that rate? Because the bacteria needs to survive, and it needs to get its sugar from somewhere. So it's getting its sugar from lactose because there's no glucose in the environment. Let's think about it the other way. If there is a lot of lactose, okay, but there is also a lot of glucose, that means that there's very little CAMP. If there's very little CAMP, very little amount of molecules are going to activate this catabolic activator. So there's not gonna be a lot of activators present in this site. That means that this transcription is not gonna be activated. Transcription may still occur because there's lactose present, but nothing is pushing that RNA polymerase to go faster. So transcription is gonna go super slowly. The enzymes produced are gonna be very few, and the organism, the prokaryote, is gonna know that they need to take their glucose, or their sugar, from the glucose present in the environment, and not use the resources to break down lactose, at least not yet, not until there is no glucose in the environment. Hopefully that made sense. Let me know if you have any questions. You guys are now going to be working on reading 2 different Khan Academy assignments. Okay, so there are 2 articles. They're overviews on gene regulation in prokaryotes and gene regulation in eukaryotes. You're going to see that the gene regulation in prokaryotes is much simpler. The overview is much simpler than what I just showed you, but it gives a good overview. And then gene regulation in eukaryotes is also super simple. We need to know it in a little bit more depth. Remember, gene regulation in eukaryotes is your chapter 19. So that is something we did not get to while we were in school, but we have read it and we have taken notes on that. After you read both articles, you're going to be assigned a activity, a worksheet. That worksheet has to do with what I just talked about right now, prokaryotic gene regulation. It is also going to be about a eukaryotic gene regulation, and it's going to be at the detail that I need it to be. So you're going to need to be using your textbook to figure some of this out. And yeah, thank you. If you have any questions, let me know.